everyone. Thank you so much for coming to my talk. Mansoura just asked me if I would introduce myself briefly. So I've already revealed that I did my PhD in the same lab as Alice Rothney with Richard Callahan, and then I did a postdoc in Manchester. And then I moved down to Birmingham to work on SMALPs. So Alice had hinted at the existence of SMALPs right at the end of my PhD, and I'd been really curious about working on them. And eventually I got the chance to move down to Birmingham to Tim's lab. And I'm still there. It's been over 5 years now, which is quite shocking, really. But it's been a really great 5 years. And it was really nice to put this talk together because I went back to some older data when I was first developing this idea of the native page method. And it was quite fun to kind of go into the archives and see some of my older stuff that didn't make it into the paper necessarily, but was part of how we started thinking about all of this. So I will actually start the talk now. So I'm just going to talk today about this simple native page method using SMA, and it's largely based on the paper we published in 2019. So if you've read that, I hope it's not too repetitive, but I'm going to try and flesh it out a little bit more. So my talk is going to be split into these 4 little sections. So I'll just talk quickly about the concept of the method and the context it's in with other native page methods. Then I'm going to do 1 slide just describing the method in the briefest detail I can, because there are a couple of points that I find quite important, which dictate how successful it is. Then I'm going to go through what I see as being the applications of it and where it's useful. But at the end, there should be plenty of time for discussion. And if other people have ideas or things they've tried and want to talk about, that would be brilliant. And finally, I'm going to just mention a couple of examples of how I've used the method and questions that I think are interesting that we could address using it. So let's go. Just need to get my cursor in the right place to advance the slides. Oops. Okay. So sorry, I haven't actually attributed this, but I took this figure from a website. It was a university's website that just really simply describes SDS page, just because I wanted to point out the kind of the analogy between SDS page and what we call SMA page. So the main point is that when you do SDS page, of course, you add the SDS, which unfolds the protein and gives it a fairly uniform negative charge. And that is what allows the protein to migrate through the gel, largely in proportion to its molecular weight. So we were talking, in fact, in one of the SMALP meetings some years ago about what's in some ways one of the slightly annoying properties of SMA, which is this negative charge, because that can interfere with some of the other methods that we use. There's some feeling that that might be what alters its behavior with certain affinity resins. So we were sort of complaining a little bit about this negative charge, but as we talked about it, I suddenly thought, but hold on, that's quite a useful property, apart from, of course, its charge, which is what makes it soluble, and that gives it the amphipathic properties. But I suddenly realized that when you make a SMALP, potentially you have a protein that is already surrounded in this beautiful belt of negative charge. And since the tris glycine gels run at an appropriate pH for SMA to be charged, then potentially we could actually make use of that to get the proteins to migrate through gels. And indeed, that's what happened. So I had this idea, and I think I went into the lab the next day and just threw some protein down a gel, which is this picture here. And it's not beautiful, but it was the very first one I did. And I just think it's nice to show a little bit of the progress. So I didn't even have molecular weight markers. I just put some BSA onto my gel to act as some kind of indicator of molecular weight, because BSA does migrate under native conditions. And what we found was we did get the proteins in SMALPs migrating nicely through the gels. We were able to show that the lipid is carried along with the SMALPs, and sort of we assume, and we were later able to show a bit more definitively that the SMA, of course, is retained as well. So it is the intact particle that migrates. Yeah, okay, so that's just a quick bit of context. I wanted to say a little bit about other native page methods, because of course there are other ones that exist, and As we've done more work on this and more people have tried it, I think it's important to say it's not a complete replacement for other native methods, and I'm not trying to sort of sell that idea. Certainly for some proteins, it seems like the more traditional blue native page and clear native page do work better. Anecdotally, it seems like smaller proteins work better with those more traditional native page methods. But personally, for the proteins that I've been working on, I've found this SMA page method really useful. So just to reiterate, it's native gel method. The SMA is analogous to SDS. So the protein migrates through the gels with the lipids, and it doesn't require any additives. So of course, the other native page methods require you to add Kumasi stain or detergent. And as I'll go on to describe later, sometimes we recover protein from our native gels. So having additives which might affect the structure or function of the protein was something we were trying to avoid. So this is just the very brief rundown of the basics of the method. It's all in the paper, and I tried to put absolutely everything into the paper so it would be easy for people to repeat. But the way that I run these gels is I use just very standard tris-glycine gels. So either, you Google it, you find the Cold Spring Harbor protocols page, and that gives you the recipe, or most commercial precast gels, which are tris-glycine that you can buy. It turns out they're SDS free, so that it can be used for native and denaturing applications. So yeah. The other thing I was going to say is, as I've gone on with this, I found that a 4 to 12% acrylamide gradient gives the best separation of the molecular weight markers we use. But of course, if you're working on a particularly large or a particularly small protein, it's worth considering going back to a single percentage gel, and also considering some of the other native gel methods if you find that this doesn't really resolve your protein particularly well. So the running buffer is just a very standard tris-glycine buffer with the SDS emitted. And likewise, the loading dye is very simple. It's bromophenol blue pH 8. I've said 20% glycerol, but just enough glycerol to make it sink into the well. What I think is nice is that reducing agents are optional. And if that's something that might be crucial to the way your protein functions, then you can look at running your gels with and without reducing agent and seeing if that affects how the protein migrates. And crucially, I'm sure I'm preaching to the choir here, but we definitely don't boil the samples, which I know is a little bit controversial for denaturing gels, but yeah, for native gels, it's an absolute no, of course. The other nice thing is that these gels run in a similar length of time to a denaturing gel. So I run at 150 volts just to keep everything a little bit cooler. It takes about 1 hour to run a mini gel. I've also experimented a little bit with running them in the cold room and lower voltages for longer times. Personally, I don't find that makes a big difference, but I'm working on some fairly robust E. coli proteins for the method development. So it's possible with slightly more fragile proteins or assemblies that you might benefit from just considering slightly kinder methods of running the gel, so lower voltage and in the cold room. And finally, when you are immunoblotting from these gels, which is possible, and I will show a little data slide that demonstrates that, I typically soak my gels in SDS running buffer before I transfer them onto the nitrocellulose membrane. And I find that really improves the efficiency of the transfer. And then of course you get better signal from the blots. So I think that's a really useful thing to bear in mind. And of course, at that point, adding SDS is not going to alter the result really. Whereas up to that point, if I, for instance, wanted to recover the protein, or if I'm interested in its oligomeric state, then it would be slightly sad to have added SDS before then. Okay, so hopefully that just gives you a quick rundown of the method and sort of how we thought about it in the first place. So now I want to talk a bit about the types of information, I suppose, that we can find out with it. And I've called this applications. So the first one, it's the most obvious really, is to use these gels to look at, can we determine accurately the molecular weight of proteins and SMALPs? And can we look into the oligomeric state of those proteins? So when I was putting this paper together, we were working at the time on these 3 model proteins, which are easy to make in SMALPs that cheap to have in the lab, and we could just throw lots and lots of them at the gels. And the other nice thing is that they have a variety of architectures. So there's ACRB, which is quite a large assembly when it's in its trimeric state. And then an ABC protein, SAV1866, which is a homodimer, which assembles to 130 kilodaltons. And then ZipA, which is a single transmembrane spanning helix with quite a large soluble domain relative to the transmembrane region, at least. So we also felt that covered quite a good variety of secondary structures, I suppose. And I also, I'm afraid it's not on the slide, but I do talk about it later. I also looked at the BAM complex just for something a little bit different because it's a beta-barrel protein. So it was just nice to show that the method was applicable to a range of architectures. On this slide, on the left-hand side of each panel is a denaturing gel, and then on the right-hand side, there is the complementary native gel from the same sample. So you can see in each case, we get the monomer molecular weight from the denaturing gel. And then when we look at the native SMA page gel, we can see a much larger protein, a protein band of a much larger molecular weight appears. So ACRB, the monomers are roughly 113 kilodaltons, I think. So the trimeric assembly is about 340 kilodaltons. And I think that shows really convincingly that we're seeing the trimer. Likewise with SAV, it's very simple. It's a 65 kilodalton monomer and then at around 130. So possibly it's running a little bit higher than we expect, but it's definitely in the right ballpark to be the homodimer. ZipA is a slightly more interesting situation. It always runs higher than its true molecular weight on the denaturing gels, and I'm not entirely sure why that is, but there must be some aspect of its structure that causes that. When we run it on a native gel in SMALPs, we get this really extensive laddering, and you see the same, which I'll show in a subsequent slide, with Western blotting. So it's definitely the ZipA, it's not a contaminant. So it forms some sort of interesting oligomeric species, at least in SMALPs. And we do see that to some extent when we look at size exclusion of the purified protein. It's quite a complex trace on the size exclusion. So that also hints at there being a number of different oligomeric species involved. So just to try to put some numbers onto this, we plotted the molecular weight markers that we use, which are shown as these black squares on the plot. And then we put onto the same graph where the retardation front for each of our SMALP proteins was. And we found that typically the proteins were running to the RF that we would expect for their size. And we just put together this chart to show there is an error if you were to calculate purely from the gel the molecular weight of the assembly and then compare it to our sort of known molecular weight. There are some errors, but probably not enough that it would really mislead you as to the oligomeric state of that assembly. So we just wanted to show that it's fairly reliable for deriving that information. Yeah, okay, so there we go. There are some errors, but they're not too big. So the next application that I wanted to mention that I personally think could be quite useful, and in fact, when I was putting this together, I found this paper that I've shown on the right-hand side here, where Sowata's group and others report that they've developed this screening method for purifying and crystallizing membrane proteins using clear native page. And I found some similar papers that were suggesting that it could be used as a sort of quality control metric. So if your protein runs nicely, if it's a single species on your native gel, then it's likely to be monodisperse and hence potentially crystallizable. And there's a lot of literature which relates monodispersity of proteins to whether they are likely to crystallize, which in the age of cryo-EM is perhaps less of a concern for protein biochemists, but it's still certainly good to know that it's out there and that my thoughts about this, that we can use the gels to monitor the quality of the protein along the same lines as other people's. So this is data also from the paper that we published. And it just compares a denaturing gel of fractions from a size exclusion run, where everything looks exactly the same apart from just the amount of protein in each well. But when we look at it on the native gel, it gives a really different picture. And this is SAV 1866. So this is our homodimer here, just in these last few fractions. And even those have a little bit of a higher oligomeric species in it. It's slightly cropped off, but these are the bottoms of the sample well. So you can also see really nicely, and it corresponds to the size exclusion trace, that where you have protein eluting in the void volume, it just sits in the sample well and it doesn't migrate in, which suggests it's probably aggregated. So if you're looking at trying to pull out a monodispersed sample from your size exclusion fractions, then these gels could be a really useful way of quickly determining which are the best fractions, in a way that the denaturing gels just flattens everything out and it doesn't give you that same level of information. So I think this could be quite useful. And I also, I've had an intention to do this, but of course things have been somewhat on hold recently, but I think it might be a useful predictor of if you store protein, whether it's freeze thawing it or storing it in a cold room, you might be able to use this to monitor its sort of propensity to aggregate, or just whether it's still of sufficient quality to use for an experiment that you might be interested in. Yeah, okay. So in some ways, I think this next application, the lipidomics, is potentially the most exciting of all, because I really increasingly feel as though the sort of unique selling point of SMALPs is their ability to directly solubilize membranes, pull out proteins with lipids surrounding them. And then if we can ask the question of what lipids are actually in those discs, that might provide us information that's really difficult to get by methods that use detergent, which are more disruptive to the lipid bilayer. So in the paper, we just did a fairly simple experiment where we compared protein extracted from a denaturing gel and a native gel. So on the right-hand side, this is the denaturing gel, and on the left-hand side, this is the native gel. We cut the bands out, obviously put them into separate tubes. Sorry, this doesn't really show it very well in the diagram, but we kept these samples separate, but we incubated them in just a standard denaturing load dye. And then we extracted from that into solvent and analyzed what lipids were present. And on the right-hand side here, you can see there is a pink line that just runs along the bottom of this mass spec trace. So essentially in the denaturing gel, there was no lipid present after we'd done all the extractions, whereas in the SMALP sample from the native page, we were seeing a lot of lipids coming out in that solvent extract. And we were able to identify different head groups and different tail lengths. And the advantage of doing it this way over just analyzing from your liquid sample is that if you do have any contaminants in your protein prep, when you extract the band just from the gel, you're really only looking at the SMALPs that are associated with your target protein. Whereas when you're with the liquid sample, if it's 90% pure and then you've got 10% other SMALP proteins in there, that could have an effect on what lipids you're reporting that you find with your protein. And I think if you're trying to get into the fine detail of lipid preferences of membrane proteins, you probably need that specificity. And this is one way to get to it. So I think this is the last sort of application slide that I have. I wanted to talk a little bit about immunoblots. Since we published the paper, we only showed this one slide. We've done a lot more, and later in the talk, I will show you some more immunoblots, which hopefully a little bit more convincing. What I think is interesting is that we use the anti-histag antibody for these because we don't have an antibody that's against an epitope within the ZIP-A protein. And what we found was we could show, so here's a pure protein that's Kumasi stained, and if you look at the denaturing gel, that is pure ZIP-A. It's from the slide I showed you earlier where it has this complex oligomeric formation. So when we blot the pure protein, we see the same bands reiterated. So they are certainly the histag ZIP-A. And we get a very similar result of multiple bands when we look at solubilized membranes. And I think that's really important because it means that if you aren't interested in purifying a protein, but you want to know something about its behavior in the membranes, or whether it's forms oligomers, whether it's part of a complex, then potentially you can take solubilized membranes and just blot those directly. And you don't have to do any purification to get to this quite specific information about the molecular weight and the assemblies of the protein. So I think that is quite a powerful thing. If you're not specifically a sort of protein purification person, but you still want to find out something about your protein, then this is a nice way of doing it. And it's almost identical to standard immunoblotting method. You just have to do that step that I mentioned of soaking the gel in an SDS containing buffer before you transfer it to actually get good signal on your blot. Okay. I told you the blotting was the last thing, but of course the electron microscopy angle was something that we got really very excited about when we were developing this. So when I was reading around about native gels, sort of probably 4 years ago now, I found this paper where they reported with soluble proteins, you could run them into native gels and then directly transfer them onto grids for EM. So in this picture, it shows it really beautifully. You run your sample across several wells. You stain some of them to actually visualize where your protein's migrated to. And then you align that to your unstained wells and extract the protein directly from there. So in this paper, they report sort of literally scratching the surface of the gel, dropping some buffer onto it, and then dropping a grid on. So this is just a tiny EM grid there, incubating it for a brief time, probably something that has to be determined for your protein of interest. And then either negatively staining those grids or potentially using them for cryo-EM as well. So yeah, these are the type of micrographs they produced from that. And of course, these are for very well characterized large soluble proteins. But I thought, given how beautifully the SMA page gels ran, potentially we could do the same thing. So that's exactly what we did. I'm showing, actually, there were two methods that we went into for the EM. In this one, we ran a gel and then we took from an unstained region of the gel, we actually just cut that band out, put it into buffer, did an overnight incubation to extract it back into the buffer, and then applied that to a grid. So this is a negative stain grid. And we got, very nicely dispersed particles, quite a lot of them. So clearly we were extracting the SMALPs and we were able to visualize them. We did a similar experiment to the original suggestion, which was directly putting the protein onto the grids from the gels. We did that with ACRB because, again, it was a large protein and we knew what results to expect. For some reason, I'm sorry, I deleted that slide from my talk, so I was worried about going too long. But it's in the paper, so if you're interested in that, you can read about how we did it and see how the micrographs actually looked. We were, with this direct blotting from the gel onto the grid, able to see very clear ACRB particles. We still haven't really progressed this to using it for cryo-EM, partly because the grid prep for cryo-EM is so much more complicated and the amount of liquid you add and the humidity and the freezing conditions are really critical. And just having the time and the access to an EM lab to get this really well defined the opportunity has not come up yet, but I would love to extend it to that as well, because I think in terms of really looking at structure, cryo-EM has to be the goal rather than just negative stain because of the resolution limit of using the stain. So to summarize, these are the applications that I see for SMA page gels. Very happy to hear if people have other ideas of what can be done. So please add questions or suggestions if you have them. But the way I see it is we can get relatively accurate mass estimates. We can see the oligomeric state of proteins potentially on the gels. We can assess sample quality. We can get precise assessments of the lipid content of discs without contaminants. It's compatible with immunoblotting methods from both solubilized membranes and pure protein. And I think that's an important point, that the membranes work too. And protein can be recovered from the gels in intact discs. So the previous slide with that micrograph, I think really clearly shows when you do extract from a gel piece back into buffer, you're still collecting intact SMALPs. And there are all sorts of experiments that you can do on those. So of course the lipidomics is one of those, and potentially EM is another. But I suppose effectively it's a sample of pure protein, and then the world is your oyster in terms of the experiments you might be interested in doing with that. And I think it might sound like a slightly odd thing to do, to purify protein, to run it in a gel and then re-extract it. But if you are struggling to purify protein using affinity methods, then potentially the gel method is another way of purifying your protein. It probably isn't applicable if you need milligram or gram quantities for the experiments you're looking at. But if you just need a small amount of pure protein, then it's definitely a route that might work. Okay, so this is the slightly newer content. I wanted to go a little bit more into what I think the useful applications of this are. And essentially, and this is something that I've become really interested in since I've been working on SMALPs. I think SMALPs could be a brilliant way to examine membrane protein complexes. Because of the direct extraction from the membranes, because we know from lots of people's work, I'm thinking of Sandro Keller because I know he joined the call just now, but the paper he published describing the sort of mild detergent-like properties of SMA a few years ago really got me thinking about this. So I think SMA is potentially a way that we can extract membrane protein complexes, and then the SMA page gels might be a way in which we can examine those and characterize them. So I started off with something quite simple, and this is included in the paper, but it's not really, I don't think we go into it in a great deal of detail. So I just wanted to mention it slightly more whilst I have your attention and we're being recorded. So this is the BAM complex. It's a complex from E. coli. It's situated in the outer membrane. It's a beta barrel protein, and it's comprised of multiple subunits. So this structure was solved a few years ago now, showing various subunits of the BAM complex. And I included the diagram just to make it really clear that the BAM-A is the only subunit of this complex which is actually embedded in the membrane. And the rest of the subunits are associated. I think BAM-B might be counted as a peripheral membrane protein, but certainly, or possibly a lipoprotein. But they're not, it's not an assembly of multiple membrane embedded proteins. Yet when you SMALP it, you still get the assembled complex. And then we wanted to sort of take that one step further and see what was happening when we ran it as the native gel. So the initial gel in which I ran BAM-B, the BAM complex, gave us this somewhat messy result, but when you look sort of carefully, you can see that it's resolving probably into 3 main bands, which I've labeled here. And then I wanted to, and that was from a pure sample of the BAM complex. So we were curious about why we were going from very clear denaturing gel with the 5 subunits shown. And then when we were running it on a native gel, we were seeing this complex picture of potentially several oligomeric states or several assemblies of the protein. So what we did was just cut these bands out, resolubilize them into a denaturing buffer, and then ran them on a subsequent denaturing gel. And this is what we found. We found that all of them contained this top band, which is the BAM-A, but there was a sort of different selection of the other subunits included in these different bands. So potentially what appeared to be a pure sample of the BAM complex consisting of all the subunits was actually a mixed population when you got sort of into the details of what was in each SMALP. So I feel like that was quite a useful illustration of just being able to look at the proteins in a little bit more detail, sort of out of the bulk solution. We also looked at mass spec of these samples, and what we found was we were able to identify all of the subunits of the complex, so BAM A to E. We also found, somewhat frustratingly, these really common contaminants that we get when we do nickel purifications from E. coli proteins, because they all have high metal affinity. But what I thought was, and ACRB is one of those, sort of usual suspects, I suppose. But we also found this chaperone SIR-A included, which is a native E. coli protein, but it is actually the chaperone that's associated with the BAM complex in its active form. So to actually find a protein that we knew should be associated with the BAM complex when we were looking at it in the native gels was something that I found really encouraging in terms of believing that this method could help us to potentially find novel interactions between proteins and complexes that are less well characterized. So leading on from that, we decided that another useful model system that did have that additional complexity was the erythrocyte membrane. So I know there's been some work published on this, possibly even 2 years ago now. The last year has gone so quickly, it's hard to keep track. But a group in France did SMALP erythrocyte membranes and showed various proteins that they were interested in as being present in the SMALPs. And I think that research is ongoing. What we were interested in doing was just asking the question of whether SMA can, generally speaking, solubilize membrane complexes. And we use the erythrocyte as a model system because the complexes that are present are quite well characterized. Once again, sorry, this is very embarrassing. I haven't attributed this diagram properly. It's from a very nice paper that describes these complexes as they are kind of currently understood. So what you can see is this protein called band 3 is a really prominent part of the erythrocyte membrane, of the erythrocyte membrane. I think it comprises something like 25% of all of the proteins in the membrane are just this band 3. There's also GLUT1, which is one of the glucose transporters, which is really important in the membranes as well. So we decided to isolate ghost membranes. So we took out all of the hemoglobin effectively because that just gets everywhere and contaminates things. So we prepared these ghost membranes by stripping out the hemoglobin. We solubilized them with SMA and then we fractionated them using size exclusion. We analyzed both the solubilized samples, so the total erythrocyte SMALP mixture by SDS page and SMA page. And we also looked at the fractionated size exclusion samples. I don't think I'm showing that data now because it gets a little bit intricate. I wanted to just give you the overview. And then we immunoblotted them to identify key proteins. And I think this is where this is interesting because I think the immunoblots from this were directly against the erythrocyte membrane proteins because of course nothing was tagged here, and the blots are much cleaner and much more sort of convincing than that anti-His one I showed earlier. So I'm not sure this is everyone's experience, but certainly my experience has been that using an antibody in these native blots that's actually against an epitope within your protein seems to work better. And then finally, in this experiment, we extracted material from the SMA page gels and then submitted that for a mass spectrometry study to try to understand if we were identifying a band on our SMA page gel, could we then ask the question of which proteins were included in that, and did it correspond to a known erythrocyte membrane complex? So here is the very sort of basic finding, which is we solubilized the membranes and this is what we got in SMALPs. So this is the SDS page and it looks strikingly similar to the many publications on the erythrocyte membranes. So up here you've got these cytoskeletal proteins, sorry, my mind went blank. So spectrin and anchorin. This big band around 100 kilodaltons, this is a silver stain, I should say. This is the band 3 protein, that's that really abundant membrane protein, and then all sorts of other proteins. When we ran the same sample on the SMA page gel, we once again got 3 fairly distinct bands, which initially we interpreted as being potentially 3 erythrocyte membrane complexes of different sizes. So we tried to look into those in a little bit more detail. We did the very simple experiment to start with that we did with the BAM complex, whereby we simply cut these bands out, solubilized them into a denaturing buffer, and then ran them into a denaturing gel. So here you can see that fundamentally the proteins in samples 1 and 2, so that's from bands 1 and 2, are the same. Lots of these proteins are heavily glycosylated, so they do have quite complex migration patterns on denaturing gels. But the third band down here, from the lowest molecular weight complex, had a completely different set of proteins in, which, from having read a lot more into it at this point, I think are one of the sort of peripheral membrane proteins, and actin down here at about 40 kilodaltons. We also blotted these samples just against anti-GLUT1, and we found that there were also two very distinct populations of GLUT1 in this sample, which was interesting because it's a denaturing gel. We would expect them all to run to the same position. But certainly neither of these proteins is the GLUT1 when you compare the migration behavior of them. And we also found, sorry, I didn't have a particularly beautiful image of it, so I'm afraid that you're going to have to slightly take my word for it, but when we did the anti-band 3 blots, both samples 1 and 2 from this gel contained this band 3 protein. So there's a lot more complexity in this. I feel like that's not necessarily relevant to this talk, which is supposed to be focusing on the SMA page. So what I just wanted to sort of round this out by saying is that when we extracted the proteins from these 3 different bands and then put them into mass spec, we got certainly 3 fairly distinct populations of proteins. There was some overlap. So for instance, SLC4A1, which is the slightly more official name for band 3, is present in most of the samples. SLC2A1 is only really present in this third one, and that is GLUT1, which we only saw by Western blotting in that third band as well. So I feel as though with this combination of immunoblotting and mass spec, we're starting to get quite quickly into some useful information about what proteins are contained in these different bands. I'm still in the process of analysing this data because it's incredibly complex and we submitted a lot of samples. But generally speaking, I'm seeing quite good overlap with the reported erythrocyte membrane protein complexes that I showed in that initial figure and the mass spec data we're getting. I think to really get to grips with it, you'd have to do quite a detailed quantitative mass spec study. But in terms of a proof of principle, I think this could be quite a useful method for sort of quickly getting some information about what membrane protein complexes we're pulling out of a bulk membrane, and also at sort of native expression levels. So rather than relying on overexpression of proteins, if we're able to do this with tissues, with samples like blood cells, then that's quite a nice system because it allows us to sort of sidestep some of the issues that come along with overexpressing proteins and then asking questions about what they're associating with and how relevant that is physiologically. So that's sort of where I'm up to in my thinking about this method and its applications. So yeah, that's that's it. I feel like I've probably spoken for long enough as well. This acknowledgement slide was too big to include pictures because so many people have contributed to this and helped, and I, of course, work for Tim Daffon in Birmingham, so my colleagues there were very helpful in terms of providing me with protein and talking things through. The mass spec work was done by Alvin, who is a student at Warwick, and Corin, and I'm particularly sort of excited about the possibilities of that work, so very grateful that they contributed that. And I also wanted to mention Vincent from Leeds Beckett, because I feel as though lots of my discussions were with him, and he is quite he's quite a stickler, and so he really pushed me to go more into more detail in some aspects of this, where I was just excited about the proteins. So I'm really grateful to him for his help. And then the red blood cell work was done in collaboration with UMass Medical School, led by Kai and Andrew, who are a husband and wife team, 2 very excellent scientists. So yeah, I think, of course, everyone on this list contributed, but those I think are the main stars. So thank you very much for listening, and I definitely would like to talk more about this. If you have experience of using the method and want to comment, then I'm really interested in that as well. Thank you.
Thank you, Naomi. Questions for Naomi? questions.
I will just stop sharing my screen for now, so that I can.
I would have a question. Hi, Naomi.
Hi, Sandra.
It's really nice. I greatly enjoyed your talk. I was particularly impressed by the erythrocyte experiments. I mean, that looks really promising. Have you tried looking at those super complexes by cryo? Because the X-ray structures of most of the individual components have been solved, right? So even though this must be a pretty dirty sample, you may be able to recognize some super complexes and even like kind of fit in the X-ray structures of individual proteins.
It's not something we've tried. I would absolutely love to, and I've been trying to write this work up recently, which is why I've been in the thick of the mass spec data, and it's made me think of all the really exciting things we could follow up with from this. Yeah, I think especially after that paper was published on the super complex from or complex, I always forget which one it is, 3. A couple of years ago. Yeah, I think that's a huge area that we should be excited about.
It's really great. Thank you.
I think there's a question from Alice who says, do we always use nitrocellulose? I've only done this with nitrocellulose membrane, partly because I'm just in the habit of always using that. I think somewhere in the back of my mind, please correct me if I'm wrong about this, I think nitrocellulose is better for larger proteins, and I've always worked on quite large proteins, so that's just my go-to. So yeah, I'm sorry, I can't answer that really. And then the native polymer page process. So I think I haven't tried this extensively. I think SMI runs quite poorly on denaturing gels. And I think the experience, there's a student in the lab who's doing some work on this, and I think her experience is you cannot get SMI to run. I mean, you reverse the pH of everything, and you just can't get it to run nicely. So I think that might be a difficult one to crack. DIBMA though definitely works, which is exciting because it's a nice alternative to have. Okay, Carrie says she's used PVDF. Thank you. Okay, great. I mean, it's really exciting to know that you're actually trying this out and working on it. I've been in touch with Carrie and Swishing a little bit, but yeah, it's good to know it's actually being useful.
Other questions?
Hi. I want to ask for the marker of the SMA page. So how do you normally stain it? You cut that part of the gel and stain with silver stain?
Do you mean in general terms, or when we're trying to look at the EM sort of application?
No, like in general terms.
So I think as long as you don't overload with marker, then using silver stain is fine. I think some people cut off the markers if they're silver staining a gel, if they're pre-stained ones for denaturing gels. But yeah, I think as long as you don't load too much marker into the lane, it doesn't sort of completely blow out when you silver stain it. Is that what you meant?
Yeah. Does the markers transfer to the membrane? Oh, no.
So I probably should have mentioned that. They may do, but because they have Ponceau S stain in them and they're really hard to see. So I think it's hard to say. And then none of them sort of, whereas some of the standard markers you use for STS page will react with antibodies and you'll be able to visualize them. Certainly with the systems we use, HRP, you often see some of those markers. So I don't have a marker that's actually usable for Western blotting. What I normally do is kind of quite nicely spaced from my native samples. I'll run a sample of an STS page marker that's pre-stained, so you can see that it's transferred to the gel. And then if you run the native marker and the STS page marker on a gel and stain them with Kumasi, then you can sort of from that extrapolate, will these bands line up? Because the STS page markers don't run true to size on the native gel, but you can do a little workaround by having a separate gel where they're stained and you can compare the migration. it would be really nice to have better markers for native gels, but maybe that's a business idea for the future.
Thank you.
Okay. I mean, have you tried that yourself? Or I know Carrie's here as well, I think. Have you tried using other markers and had a slightly more efficient experience than I have?
I've just been using SDS page markers, stained ones for the blots, the same as you. So
okay, great.
I haven't really, I don't even have the native mark ones yet. So
okay. Yeah, I find the native markers really useful. They can be quite hard to interpret at first because I don't know, because they're not stained, I suppose we're so used to looking at those kind of pre-stained markers, it can be quite hard to line up the molecular weight with the sort of the literature you're given when you buy the product. But once you get used to it, it's really easy to see which band is which.
yeah, I guess I found it useful to have something so that you can kind of line up different things that are run on different blots. So you can at least kind of line up to see,
yeah,
because I'm trying to see if different things are in the same SMALPs or not. And so at least I can line up the regions of the gel and say, yes, this, you know, this protein's in the same area of the gel as this protein. And so suggest they're in the same.
Exactly.
Yeah.
There's certainly a lot more work to be done on this and a lot more sort of precision and detail that I think could be added to the method. But yeah, it's hard to know what the questions are and like what the challenges are, I suppose, until people have used it a little bit and kind of come up against them.
Can I ask another question?
Sure, go ahead.
Sorry. So I want to ask for the bands that represent oligomers on the SMA page, like you see the higher bands. How do you make sure they are oligomers or they are aggregates of SMALPs? Do you just think aggregates doesn't like aggregate into the gel?
I think, yeah, that's a good question. I suppose in some ways it depends on where you draw the line between what's an oligomer and what's an aggregate. So when we run fractions from size exclusion, you can definitely see a difference between protein that just doesn't even migrate into the gel and then high molecular weight bands. And I think I would probably distinguish between those two as being some oligomeric form versus just a fairly random aggregate of protein. The question is, of course, whether those oligomers are truly representative of the protein in the membrane or whether they are some artifact of the SMALP, which I know is not a helpful thing to say, but I think it's an honest thing to say that you have to keep that in mind, that we know that certainly SMALPs do kind of self-interact to some extent. I suppose that's why there's a lot of work being done on alternative polymers to try to overcome some of those difficulties so that we can really be a bit more certain about what we're observing. Yeah, I suppose the other thing is, as Sandra mentioned, you know, if you look at these things under EM, then if you're seeing random assemblies of things, then it's more likely to be an aggregate. Whereas if you're seeing ordered assemblies, it's probably more likely to be real.
Thank you.
It's also good to like add some GFP, RFP, YFP to protein and just look at the, I mean, just look at the, after running the gel, the native gel, just look at the color of the gel, how it looks like. Is it yellow? Is it like a mixture of yellow and green or green and red? Something like that.
Oh, I see. So, oh, that's really, yeah, okay, that's really interesting. Do you mean co-expressing different proteins with different GFP tags or mixing different samples which have different GFP SMALP proteins?
And like nanobodies that are conjugated with GFP, RFP, YFP, something. If you have antibody for your protein.
Yeah, I guess that's always the question. Yeah.
Excellent. Nice discussion. SCS page is one of those methods that we use a lot in the lab, so a lot of questions came around. Excellent.
Yeah, I think the nice thing for me about this is it's just such an easy add-on to STS page that it doesn't require any different equipment, any different buffers to speak of, or at least the buffers are things you can really easily make for yourself. So it does make it quite a kind of low bar to entry in terms of trying it out, which I think is helpful, definitely.
Do you think this can be applicable to, for example, DNA nanodisk? You know, that's.
I suppose it's possible. I guess it just depends on the charge and the pH, you know, the buffers that you're using and the charge on whatever is the sort of the belt of your disk, if you like. I did notice sort of retrospectively that these proteins that we tested did all have PIs that would make them negatively charged under the conditions that we were running the gels. So it's possible that that contributes to whether a protein migrates nicely and gives you sharp bands. So if you're kind of fighting between the charge on the SMA and the charge on the protein, it might not work so well. But I think finding a kind of a range of proteins that you can get into disks and you know make enough of and test, it's quite a big job. And I suppose that's why, as you test it with different proteins where you're specifically interested in the protein, I think little kind of variations and problems or successes hopefully will emerge. But it's hard to kind of predict them all in advance, I think.
So thank you so much. So anything that you want to add?